Good. All right, thank you. I want to introduce Phil Bergmann. He is completing his third year of graduate school. Just for some background, Phil finished his undergraduate work at University of Minnesota. After that, he went to Prevention Genetics for 5.5 years, where he worked in the R&D department. And then he came here and 1921, a long time ago, and just after the first pandemic. It is. In 2021, he came here and he joined Biolab, and he's going to tell us a little bit about his work for his thesis project, which is focused on the sex-specific role of DNA methylation in Alzheimer's disease. Phil.
Okay, thank you, Reed. I guess let's just hop right into it. So just a brief outline of what I'm going to talk about today. We'll start out with some background on mild cognitive impairment, Alzheimer's disease, DNA methylation. We'll highlight some of our recently published data in this field, move on to the sex-specific DNA methylation, and then just a brief slide on kind of the future directions with this work. So background, I don't need to explain Alzheimer's disease too much to you guys. We're all aware it's a growing problem in the US and throughout the world, with cases in the US expected to more than double by the year 2050. And this is leading to astronomical healthcare costs expected to exceed over $1 trillion a year at that point. So my thesis work is focused on studying the progression of Alzheimer's disease. So we expect 10 to 18% of adults over the age of 60 to transition from cognitively unimpaired to mild cognitive impairment, which have declines in memory, language, and executive function. And then 80% of those individuals within 6 years will convert into Alzheimer's disease. So Alzheimer's disease is characterized by progressive neurodegeneration, leading to loss of neurological functions. Symptoms progress all the way to loss of independence for the individual. It affects 1 in 9 adults over the age of 65, with 2/3 of those individuals being females. And so while the exact molecular mechanisms behind Alzheimer's disease is not known, there are several molecular etiologies that have been identified, such as tau neurofibrillary tangles, beta amyloid plaque formation, and neurodegeneration. And so it's also important to just note right now, there are kind of 2 different types of Alzheimer's disease: early onset Alzheimer's disease and late onset Alzheimer's disease, which I'm abbreviating as AD throughout this presentation. And it's what we study. It accounts for 90% of the cases. Your symptomatic age is over 65 years, and its genetic basis is unresolved. But several genetic risk factors have been identified. So the best known is ApoE4 genotype, but TOM40, BIN1, and CR1 have also been identified as risk factor genes. A recent GWAS study also identified 75 risk loci associated with AD when looking at 800,000 people. And this leads to a heritability estimate of about 60 to 80%. So as that might tell you, the Mendelian genetics does not fully explain AD incidence. And so our question was kind of, does the environment influence the development and progression of AD? And so this first case report identified 55 monozygotic twins that were discordant for AD in the Swedish twins registry. To kind of get at what might be different between those twin pairs, this group looked at brain tissue in 2 pairs of monozygotic twins and identified global demethylation in AD. And then kind of even going further, this last study looked at 23 discordant monozygotic twin pairs and identified 11 differentially methylated regions in blood. So that kind of brings us to what exactly is DNA methylation? Well, it's the most commonly studied epigenetic modification. It's a covalent modification of a methyl group onto cytosine, typically within a CpG dinucleotide, so a cytosine followed by a guanine. It is modifiable and influenced by the environment. It's variable throughout your life and alters gene expression. So in this simple diagram, you have an unmethylated promoter, which allows transcription factors to bind, and a gene transcription, whereas if that promoter is methylated, the transcription factor can no longer bind, and you don't get any gene transcription. And so this is another one of the mechanisms in which case we can have a diverse number of cells throughout our body with just one genome. And so how do we detect DNA methylation? So most studies up until this point, including all studies in Alzheimer's disease, have utilized methylation arrays, which look at only 450,000 or 850,000 sites throughout the genome, which is less than 4% of all the sites. So our lab is focused on using whole genome methylation sequencing. And with that, we get over coverage of over 25 million CpGs. So our lab investigates easily accessible tissues such as blood to identify episignatures of disease. So we can take a cohort of individuals with a specific phenotype. We can extract blood, isolate DNA, perform whole genome methylation sequencing, and ask if by analyzing the DNA methylation, can we stratify this cohort into their phenotype? And so the goals of this are to improve detection and prognosis, improve our understanding of individual pathologies, and identify novel therapeutic targets. And so our lab works with human subjects. And so just take a brief second to mention some of our considerations that we face on a daily basis. So first and foremost, having an IRB approved protocol. Kind of main points of that are making sure all of our participants have informed consent, and then as well as us researchers having the proper training. So that includes city human subject training as well as HIPAA privacy training. For data access, so we only have one collaborator that can access participant medical records. When in our day-to-day work, all of our participants are de-identified, and we limit the amount of protected health information to just what is needed for our analysis and to answer our questions. Our data storage is done in HIPAA compliant controlled access, such as Silo, our own server, and moving files using Globus. And then when we publish the data, we publish it in an appropriate database, such as dbGaP. So that brings us, I guess, to our first question. So are there DNA methylation differences between AD, MCI, and CU in blood? And so we can kind of get at this question with kind of three different pairwise comparisons. So MCI versus CU, basically, are there DNA methylation changes associated with MCI? AD versus CU, are there DNA methylation changes associated with AD? Or AD versus MCI, are there DNA methylation differences between MCI and AD? Just looking at our study demographics, so all of our participants come from the Wisconsin Registry for Alzheimer's Prevention and the Wisconsin Alzheimer's Disease Research Center. We have a total of 382 participants with 174 cognitively unimpaired, 99 with MCI, and 109 with AD. They are all roughly matched for sex. And then we also have several other covariates such as age, education, and body mass index that we can also use in the analysis. So we performed whole genome methylation sequencing from whole blood using the EM-seq method. Again, we have coverage of over 25 million CpG sites with an average of 561 million uniquely mapped reads per sample with an average read depth of 38x. Our statistical modeling accounts for covariates such as white blood cell composition, age, sex, and BMI. We do perform a multiple test correction. And then to define a differentially methylated position, or as I'll probably refer several times in the future, a DMP, we required an adjusted significance of less than 0.05, as well as a methylation difference between groups of 2.5% or greater. And so what does this all mean? And so this is just looking at one shot in the genome. This is specifically in the AD versus CU comparison. And so I'm going to be showing a lot of these UCSC figures moving through. So I'm just kind of going to walk us through how to kind of read this. So at the top, we have a scale bar and then the genomic coordinates right below that. So in this case, we're looking at about 600 kb on chromosome 12. Below that are the illustration of all of the differentially methylated positions. So each lollipop represents a differentially methylated position, with the white being hypomethylated positions, in this case, there's less DNA methylation in AD participants compared to cognitively unimpaired, whereas the black or filled in circles are hypermethylated positions, in which case there's more methylation in AD participants versus cognitively unimpaired. The height of the lollipops represents this adjusted significance, and then these little tick marks along the bottom are a 1 to 25 downsampling of all of the non-significant sites throughout the genome. Below that, we have a canonical transcript of genes shown, and then even further below that is at the bottom are in green are CPG island designations, which are dense clusters of CPGs believed to be involved with gene expression changes. And so kind of just looking at this, we have a hypomethylated position in IPO8 near the promoter region of that gene. So this gene is transcribed. If you can see the little chevrons there, it's an antisense gene. Whereas if we move on to T-span 11, we have a hypermethylated region or hypermethylated position at the 3' end of the gene. DDX11 has hypomethylated positions at both the promoter region as well as the 3' region of the gene. And then we can also see that there are several intergenic differentially methylated positions downstream of DDX11 and right in the middle as well. So we can also visualize this in the form of a volcano plot. So again, x-axis is going to be the methylation difference between participants with AD minus the average methylation of the cognitively unimpaired participants. So on the right side are going to be hypermethylated positions in red, and on the left side are hypomethylated positions in blue. And so we can also then perform the same analysis for all three comparisons. So I showed you the AD versus CU. So now adding in the MCI versus CU and the AD versus MCI comparisons, and we find 10,000, 15,000, and 20,000 differentially methylated positions in each comparison, respectively. One other thing I'll point out here is in the MCI versus CU, the majority of the DMPs tend to be hypermethylated, whereas in both the AD versus CU and the AD versus MCI comparisons, the majority are hypomethylated. Next, we can annotate all of those positions to genes, and then also can kind of compare between all of these. And we actually find 392 genes that are differentially methylated in all three comparisons. And so we ask kind of is there any relationship between those genes by performing a gene ontology analysis. And we find numerous neurologically related terms that would indicate functional relevance for Alzheimer's disease, such as synapse organization, synapse assembly, and neurotransmitter secretion. We also ask, is there a potential biomarker potential with these identified DMPs? And so we utilize a machine learning algorithm to rank our top DMPs. And then we performed clustering on those top DMPs. And we're actually, with 19 DMPs, we were able to actually differentiate all three groups from each other using the methylation data. So in summary of this first portion, we generated whole genome methylation sequencing for 382 participants from whole blood with an average coverage of over 25 million CPG sites. We identified numerous differentially methylated positions, including in promoter regions, potentially leading to gene expression changes. And our top candidate positions were able to differentiate AD, MCI, and CU. So I guess kind of moving forward again, as I mentioned at the beginning, there is a sex bias in AD. So that brings us to the next question is, are there sex-specific DNA methylation patterns found in AD, MCI, and CU? So we can use the exact same model setup, except now we have 6 different groups instead of 3. So now when we do these comparisons, it's going to be male AD versus male CU, female AD versus female CU, and so forth. And so again, we do the same multiple testing correction. And so to actually become a differentially methylated position, we require that significance to be less than 0.05. And then in this case, I'm requiring a methylation difference of greater than 5% between groups. And so again, on the right is the same volcano plot, except in this case, in red are the female differentially methylated positions, and in blue are the male differentially methylated positions. And so that once we have this plot, now the question is, how many of these positions are actually shared between males and females? And so just kind of a simple Venn diagram, we actually find that males and females are almost completely distinct from each other. So we only have 47 positions that are shared between males and females in this specific comparison. Again, we can apply that to all our other comparisons, so MCI versus CU and AD versus MCI, and that same trend holds true, where we only find 136 or 139 differentially methylated positions that are shared in MCI versus CU, and only 95 in AD versus MCI. Again, indicating that males and females are almost completely unique in regards to the differentially methylated landscape. Now we can also look at within sex. So if they're distinct from each other, are females or males within these three comparisons, are there any overlaps? And again, we find that there are very few overlapping differentially methylated positions in all three comparisons, with 13 and 14 for females and males. Again, indicating that females and males are very distinct at the DNA methylation level, as well as each of these different categories is also very leads to distinct changes in the methylome. Next, we can annotate all of these differentially methylated regions to genic structures. So again, these graphs are kind of going from 5' to 3' in regards to like kind of a gene structure. So we have 1 to 5 kb upstream, promoter regions, 5' UTRs, exons, introns, 3' UTRs, and intergenic regions. The y-axis is the percentage of DMPs annotating to each of those regions. We have 3 bars for each section, so female, male, and then all CPGs, which is taking all 25 million sites and seeing how they would annotate proportionally as well. And then for males and females, we have split bars indicating the number of hypo, or the percentage of hypo and hyper methylated positions. And so I'll just kind of point out that the vast majority of the differentially methylated positions that we find reside in introns or in intergenic regions. Again, we can look at this for all 3 comparisons, and we can see the same trend holds true with introns and intergenic regions accounting for the majority of the differentially methylated positions. We can also see, similar to the earlier data I showed, that in the MCI versus CU, we see an increase in hypermethylated positions, whereas in both the AD versus CU and AD versus MCI, we see a shift to hypomethylated positions. So next, let's look at some more genes and see how kind of some unique scenarios that we can kind of come across. So again, looking at the gene TRIM71, the difference with this plot now is I have the female differentially methylated position track on the top with the male differentially methylated positions track below it. And so here we can see in females, we have a cluster of 30 differentially methylated positions right near the beginning of the gene by the promoter and exon 1, as well as another downstream differentially methylated position. But then when we look at males in the same region, we see no significant positions at all. But we can also see the opposite. So looking at a different gene, NR2F6, now females have no differentially methylated positions in this region. But if we look down at males, we see all a cluster of 34 plus a couple downstream DMPs, again, this is an antisense gene, so this is right near the beginning, also falling within the CPG island as well. We can also find positions, again, with those Venn diagrams I showed, there are several positions that do overlap. So if we zoom in here in the PPP5C gene, and we can see 3 positions that are the exact same between males and females, and they're also methylated in the same direction. So in this case, they are all hypomethylated positions. And then the next choice is obviously the opposite. So in this case, the gene EVC has a differentially methylated position, where in females it is hypermethylated and in males it's hypomethylated. Again, indicating that there could be gene expression differences between males and females. Again, we can annotate all of these differentially methylated positions to genes, and the same trend kind of holds true that we saw earlier, that for the most part, these genes are still very unique between males and females, with only having an overlap of 157, 154, and 357 genes in this comparison with significant numbers of sex-unique genes. And so again, we can ask, like, what are the relationships between these 3 sets of numbers? So kind of going through step by step with this first comparison. So again, we're looking at the AD versus CU comparison. Shown is a gene ontology of the overlap genes. So the 154 genes are shared between both males and females. And again, we find an enrichment for a lot of neurological related terms, such as dendrite development, neuromuscular process, and synapse organization. We can perform the same analysis, again, though, using now the male-specific genes, and we find only 3 gene ontology terms related to cellular adhesion. Moving down, obviously, the next obvious choice is then to look at the female-specific differentially methylated genes. And again, here is where we see a further enrichment for neuronal-related genes, such as axonogenesis and regulation of membrane potential. So again, we can kind of continue with our 2 other overall comparisons in MCI versus CU, and we see the same trend here. Even the main difference here is the male terms resulted in no significant gene ontology terms. And again, for AD versus MCI, a similar story as well. And so lastly, I just want to show a little bit of kind of maybe teasing out a little bit of function. So we were wondering if there's any way we could kind of identify like what these intergenic differentially methylated positions are doing. And so what I did, I took essentially, I have 6 lists of DMPs. And so basically I asked which of these fall within a blood-specific promoter capture high C data. And so with that data set, we have enhancer regions throughout the genome that are blood-specific. And then as another filtering step, I then asked of the DMPs that fall within those regions, how many actually fall within ENCODE designated enhancer regions as well. And so we get some modest numbers between 38 and 164 differentially methylated positions for these comparisons. And so I think in this case, a picture is worth 1,000,000 words. So looking at PDIK1L, the gene is at the top left over here. So this is the promoter bin in the, and so this large region at the top. So this is visualizing the promoter capture high C data set. And so the loops indicate the enhancer regions. So there are 2 enhancers shown, and then there are 4 more enhancers that are further downstream. And so below that, I have my differentially methylated position track. And so we can see there's one hypermethylated position located in this enhancer. And then at the very bottom are the list of the ENCODE candidate cis-regulatory elements. And we can see that that falls within a designated distal enhancer. And so in summary, DMPs are predominantly distinct between males and females in all 3 comparisons. Female-specific genes reside in biologically relevant pathways, giving insight into sex differences in AD, with the majority of our DMPs falling in introns and intergenic regions, which may play a role in the 3D genomic landscape. My immediate next steps with this data is kind of identifying top candidate DMPs for prediction, as well as validating some of these positions as well. Just a little bit on future directions. So using this data as well. So traditionally, DNA methylation analysis excludes chromosomes X and Y, and that's because of X chromosomal inactivation, as well as copy number differences of the Y chromosome. And so we have a large enough data set here, we can sex segregate our cohort and perform both of these analyses as well. We can also, since it's whole genome, we can look at all cytosine residues within the genome and look at CPH methylation analysis. So cytosine methylation that's followed by any of the other nucleotides besides guanine. And then kind of a little different take is looking at methyl QTLs. And so that's the goal to identify genetic variants which are correlated with changes in DNA methylation at specific sites. So in this scenario, you have an A allele, which leads to methylation downstream. When you have a G allele at that position, the downstream region is unmethylated. And so this can give some functional relevance to intergenic G loss variants. And with that, I just want to thank my lab, Dr. Alish, and all of our lab members, our collaborators, as well as my committee. And I can take any questions now.
So you showed nicely that your male and female data are quite different.
Yes.
I'm just wondering whether all individuals are different. And that if you took your male population, randomly split it in two, and compared those two groups to one another, would the overlap be more significant than what you saw between males and females, or would it be the same?
Oh, that's a good question. I don't know if we've necessarily looked at that. Like, this is definitely like all of this data is just a snapshot at times. So your DNA methylome is changing constantly as well.
Right.
So I guess I would hope that they would be like fairly similar, but there's no way of yeah, that's I think a good kind of gut check to perform.
Well, yeah, I think it says something about, the population as a whole, is it a sex difference or is it just a general difference among the population?
Yeah.
Maybe there's better ways to do it, but that's what I usually.
Yeah, it's an interesting thought for sure.
So I was curious about how genes involved in neurons are enriched in these white blood cells. And I'm wondering kind of to what extent is the methylation pattern within one person different between different tissues? And if you think, do you imagine that there's like an endocrine signal that's coordinating methylation within a person between their neurons and the blood cells, or maybe these genes are annotated for function in neurons, but they're really like generic cell biology effectors and they're functioning in blood cells too?
Yeah, so it's a great question, and we receive a lot. So for sure, cell types have differences in methylation. That's given, but our postdoc in our lab actually has looked at in mice brain tissue and blood, and I think it's about 60 to 70% overlap in differential methylation when looking at those two different tissues as well. So I think there's definitely some case. I also think with like kind of when I talked about the methyl QTLs as well, there are regions that are kind of more genomically influenced versus environmentally influenced as well. And so I think that's one of the exciting parts of the methyl QTL analysis is that those differentially methylated positions that we find with that, we would predict to be more consistent across tissue types than some of these other positions as well. Does that answer your question?
All right, so are you saying that that methylation pattern is genomically encoded?
I wouldn't say like genomically included, but definitely influenced more so. So there are several studies that look at methyl QTLs. And they find they're abundant throughout the genome, just kind of linking these variants to methylation. There was also another paper that identified they're called corsives. So they're correlated regions of systemic inter-individual variation. And so what that basically means is between people, the methylation at these sites is very different, but within a person, the methylation in different tissue types is very consistent. And so that's actually kind of another future direction. I actually kind of want to overlap my data with those defined regions and just see, because that in the end, yes, we would love to have brain tissue, but that's very difficult to obtain. And so if we can kind of tease out through these other methods, potential sites that are changed in brain, I think it would be really interesting.
Can you assess, my question, can you assess the viral load in these patients? Because viruses are known to hypermethylate genes and also perhaps induce change, cause RNA condensates to form, actually.
Yeah,
Which may be suggestive of the disease.
interesting. We should be able to, I think. It is, so I guess typically most researchers when doing this do bisulfite conversion of all the DNA. We use enzymatic conversion, the same, in the end, the same result. So I guess I don't know. I haven't heard of people doing alignment, essentially just trying to align reads to viral genomes. I think it's a definitely interesting thought to think about as well. Dr. Worling.
Hi, so really great, interesting project. You showed the breakdown of differentially methylated regions across basically regions of a gene, right? You show intergenic regions and chronic regions.
Yep.
And the proportion of sites in each of those regions roughly mimics like the distribution of the human genome writ large, right? Have you looked at whether the number of differentially methylated sites in each of those subregions is more or less than expected, given the makeup of the human genome?
It's on the to-do list.
Okay.
Currently hasn't been done, like kind of swimming in data at this point, but for sure we are checking for significance of those spots.
I'm not super familiar with the analysis. So do you also do just whole genome sequencing so you can correlate that the changes in the methylation aren't due to an underlying change in the sequence or deletion? Like if there were if there are regions that tend to vary between individuals, you would maybe pull that out because of the sample size. And I don't know how.
So currently, no, we don't
Okay.
do side by side, but we actually can do variant calling from methylated sequencing data, which is how we'll do the methyl QTL analysis.
Okay.
But
in your model, you show it as some, this site, but not underlying site. But you could imagine that there would just not be, there would be a deletion of that site.
Yes. Correct. Yep.
And then also, have you shown the distribution on chromosomes? Like, so my immediate question was whether you excluded the X, right?
Yep.
And the Y for the reasons you mentioned. But then I guess my question is, have you ever just like plotted the differential methylation across the, we often look at, see if.
If like one chromosome is enriched
For
for.
certain domains, certain areas tend to be.
Yeah. I feel like I haven't done it lately. I think I might have done it right away and I don't recall anything being super significant. Yeah.
For the genome-wide hypo and hypermethylation patterns, it looked like MCI was not intermediate between AD and controls.
Yeah, that's what we're finding. We're almost kind of thinking it goes up and then crashes down at AD. Again, we need to confirm this in other cohorts and whatnot. But that's kind of our running hypothesis right now. And kind of just also telling us that like these states are very distinct from each other, even though MCI is kind of, it's a much more flexible state. So patients, when they're diagnosed with that, they can actually exit back to cognitively unimpaired for time. And so it's just a really interesting kind of group of individuals to look at.
So when you split the population into males and females, and then you further look at the overlap between groups, groups, and you found like 13 for males, 14 females, did any of those overlap between?
Yeah. So like a grand, like, so I didn't show it, but like I actually made like a 6-way Venn diagram, which is a nightmare to read. But it's 3 or 4 positions that overlap all comparisons. If I recall, they're like located near the centromere on chromosome 4, not near any gene. So unknown significance, but there are positions that overlap all comparisons.